Hello everyone out there in YouTube TV land. So please, I think it's the micro lecture, and I'm waiting for comments to come in on Molec and Cell, but already there's a comment about someone that had their herpes and depression cured by a doctor, and I think it's a wonderful website you should probably go check out, but no. I'm glad you're back. So you were never in immediate danger, or?
Well, I mean, I studied a test with this girl that's on my team that did test positive for COVID. So then I tested again, like 4 days after my window, and I was negative, and then I tested again yesterday, and I was negative.
So you're negative.
And nothing.
Yeah, so apparently that's still the rough one.
Good. Let's see. So hopefully everybody's watching the movie and are down with what we've been talking about in terms of the movement of molecules across the plasma membrane. Oh, before I forget, the syllabus has an exam set up for like at the end of this chapter, and I'm like, no, I'm not sure how that got in there. There was a time where I used to try and cram 3 exams before the final, and I'm like, that sucks. So we're going to cover metabolism, and then we're going to have our exam. And so that means that we got next week of the stuff to talk about, and so it'll probably be the week after that we're going to do an exam, probably the same way we did this again, where it'll be a 2-hour thingamabob. But I'll start posting sort of the chapter summaries and stuff as we get further along. And there you go. So let's see, lab. This week we're doing the polymerase chain reaction, PCR stuff, and counting up colonies. It's a short enough lab that we have time to actually talk about things in class and see how that goes. And so we've been talking about the nervous system. And specifically we've been talking about some of the different kinds of transport mechanisms. And we can break things down into passive transport, which is using the diffusion gradient, and molecules are moving from an area of higher concentration to an area of lower concentration. And we can categorize passive transport in a couple of different ways. This idea, what's known as facilitated diffusion, meaning that we have some kind of protein channel that is facilitating the movement. Because anything with a charge, any ion, and most metabolites, glucose, all the different amino acids, anything other than a nonpolar molecule has to go through some kind of pore or a channel. And when we start talking about hormone signaling, we'll see one of the key differences in hormone signaling is steroid hormones can diffuse across the plasma membrane. So you can actually apply a steroid hormone to the skin surface and it'll actually diffuse into the cells. Whereas a peptide hormone, a hormone made with a protein, it has to have an external receptor. And so this diffusion biz, is an important topic. So we can have, in terms of passive diffusion or passive transport, we've got just the diffusion of water, which may or may not require aquaporins. That's kind of like a bit of a contested topic, I think. And also the water can diffuse through the membrane itself. Then we've got these 2 different kinds of facilitated transport systems. The idea of the transporter, which really is binding to the material that's passing across the membrane, and it's interacting and kind of changing shape. Consequently, it ends up being saturable, and it ends up being very specific. So the idea of glucose, we've got D-glucose, the metabolically active form of glucose, is able to interact with the glucose transporter, but L-glucose doesn't work. And then we also have the idea of the channel, or the different kinds of, basically a tube. And so these are controlled. They've got regulatory systems, like a ball on a chain that is sticking in the hole, and then a signal can come through, like a voltage spike, and cause that ball to now drop out, and that channel opens up. So we have all these various kinds of ion channels, and the nervous system is a really good place to put them into context, because every ion channel regulatory system that we can imagine is present in the nervous system. And we went through a little bit that we have the ligand-gated ion channels, which we're about to get to. We have mechanosensory-gated ion channels, which are things like stretch receptors and sensory neurons, like for touch or for hearing. They respond to pressure or some kind of movement. And then we had the voltage-gated ion channels, and these respond to a change in voltage. So the cell, if we kind of think about this, and it actually kind of carries over into our metabolism discussion, the cell can be viewed as a little bit of a battery, in that a battery works because you have an asymmetrical distribution of electrons. In this case, we have an asymmetrical distribution of charged molecules. We've got a lot of sodium on the outside with a positive charge, and very low sodium on the inside, and a lot of negatively charged ions, a lot of negatively charged macromolecules, all these proteins and sugars. And so we have a charge differential that actually generates a very small voltage between the inside of the cell and the surrounding fluid. And this charge differential is something we can actually take advantage of. And so the neuron is able to transmit its signal by transmitting the voltage change down the cell. We've got the cell body here that is taking signals in, and as we'll see in a moment, this is where the excitatory or an inhibitory signal starts, in that we have sodium rushing into the cell, which is going to change the overall charge. So we have a sodium channel that'll snap open. That's going to depolarize a little portion of the membrane. And as it depolarizes, it causes the next adjoining portion of the membrane to now depolarize. And you have this sort of, like knocking over a series of dominoes. And because you hit this one, and that starts the dominoes falling, the voltage-gated ion channels open this way, the ion mix in the cell is completely messed up, and so you can't have the voltage change running backwards because this immediate cell has just depolarized, and once it actually undergoes repolarization, it actually has to reshuffle the ions around for a moment, and so it takes a few moments for this patch of membrane to be able to actually repolarize again, or this patch of cytoplasm. So it's not a single entire cytoplasmic pop. And, it's because this stuff actually does take a small amount of time. Embryos or eggs, when they get fertilized, actually have a very fast reaction with the release of calcium. And there's a calcium wave that flows across the now fertilized egg. And this is part of the activation process. And this flow of calcium happens because the sperm hitting the cell surface of the egg, burrowing through it, and then once it makes its way inside, it snaps and opens up calcium ion channels that flush calcium across, in a wave across the embryo or the egg. And in the same way, we've got this movement of ion changes moving across the neuron. And so the idea is it doesn't happen in the cytoplasm as a whole like that. It happens in these patches and moves down. So the main point is we've got a higher concentration of sodium outside than inside the neuron. This is the standard way we maintain our cell ion concentrations. The voltage-gated ion channels open up, or sodium channels open up, change the sodium concentration, cause what we call the depolarization of going from minus 60 millivolts to plus 40 millivolts. And we have this peak. And this change then causes the potassium channels to snap open. And potassium, remember, has been accumulated in the inside. It starts to leak out. That now begins to drop this thing down. We actually go through what's known as an overshoot, where we go below minus 60 millivolts to about minus 70, and then the sodium-potassium ATPases are kicking into high gear. They start pumping the sodium out, potassium in, and we reestablish the normal ion concentrations in this portion of the cytoplasm. So this continues down, 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 until we get to the end. Now, one of the things, and it's a actually a really good control mechanism, is that the end of the neuron, which we refer to as the axon, doesn't physically have a connection with the downstream or the post-axonic tissue, which could be a neuron or a muscle. You actually have a little air gap. And one could imagine, like if you're, wiring up a little computer or something, you would put a drop of solder on the wire to make a connection. Or you'd physically take the two wires and wrap them together and put a little bit of electrical tape on it. So you have a physical connection here. In the case of the cellular systems, we actually have an air gap, or a fluid gap, in that the axon doesn't have the ability to transmit the electrical impulse to this downstream cell. Another thing we'll see is that plasma membranes are actually fairly insulative. That phospholipid bilayer, it's preventing ions from moving across. It prevents the flow of electricity. So cells are reasonably insulated from each other. Now, if we do sort of a high mag of what's going on here at the axonal junction, we can see some interesting effects. And this is where we can also begin to do some interesting neurological hacking. We mentioned on Monday, we have some interesting ways you can hack the neurological system. One of them is there are mechanosensory neurons, specifically the neurons that are interpreting temperature change. They, in addition to being opening because of a temperature change, hot or cold, they also, for whatever reason, respond to a small chemical. They are, in fact, both temperature-gated and ligand-gated. And so you can apply a specific chemical to the skin and mimic the feeling of dropping the temperature. And this is how stuff like Icy Hot works, or Bengay, or whatever, atomic balm, whatever is used in physical therapy, some sort of cream that you rub on your skin, and it starts to feel warm. And what that's doing is increasing blood flow, because the skin says, hey, it's getting hot right here. You start having increased blood flow, which does whatever it does. And it really is a spoof of the nervous system, because, the temperature didn't increase, but you feel warm. It's one of those interesting kind of little odd things happening. So we've got the axon coming in. Here it is. And then we've got the dendrite here. And there's another one right there. All right. So we have the axon. We have the dendrite, just the various vocabulary terms we use. And we have this air gap, or a fluid gap. This is referred to as the synapse. So another term we would use is, this is known as the synaptic cleft. And it is a fluid-filled spot. So we can now think about the presynaptic cell and the postsynaptic cell, and kind of refer to things that way as well. So the voltage is really very low. I mean, it barely is going to light up an LED. And so when the voltage depolarization gets to here, there's no way for that voltage to jump this gap. In a car, we have a spark plug, but that uses a whole lot of volts to be able to generate enough voltage in thousands of volts to actually jump that gap and create a spark. So there's no way you can get a spark going on with the low voltages that we've got in our bodies. It'd be an interesting thing to think about if you did have actual sparks going on. It could be a potential fire hazard as well. Your brain would fry. But rather than having the voltage actually transmitted in from the axon to this next cell, we've got the release of chemicals at the axon end that are then being picked up on the dendritic end. I'm actually going to draw this in even higher mag. So we're zooming in on the synapse. There's the synapse, and then we'll just go ahead and keep our dendrite right there. So we got this great big cleft. Now, when the membrane depolarization hits the end, there are a whole set of vesicles that are sitting around here at the axon terminus. And they actually are anchored into a set of proteins at the base there, part of the cytoskeleton. And there are some interesting mutations if you jack with those anchors, but regardless. When the voltage change happens and the sodium is rushing in, it causes these vesicles to release, and they then fuse with the axon terminal. So we got a vesicle fusing. This is one of those notions where the topology is important, how the way the vesicle fuses. The vesicle fuses, and it dumps the contents of the vesicle into the synaptic cleft. And of course, these vesicles contain compounds collectively known as neurotransmitters. The neurotransmitters flood the synapse and actually fairly quickly diffuse across. And on the postsynaptic end, we've got our last set of gated ion channels, and these are the ligand-gated ion channels. So we've got something that can bind this neurotransmitter. And when the neurotransmitter binds, that causes a release of, or an opening in the ion channel, and sodium can rush in, and bada bing, we have established now a new membrane depolarization event. And if this is in your brain or in parts of your spine, we have an axon synapsing with another neuron, and this is what's going on in your brain right now. Or in the neuromuscular junction, you have a neuron synapsing with a muscle. So in the neuron, you have another membrane depolarization event occurring. In terms of an excitatory signal, you have a sodium channel snap open, and that causes this wave of depolarization, and you're off the races for the next round. In a muscle cell, you have the sodium rushes in. That causes a membrane depolarization here as well, but instead of having the signal being transmitted as a membrane depolarization, it's going to cause a release of calcium. And if you recall, we were talking about the fact that calcium is one of these things that you really keep in vanishingly small amounts in the cytoplasm. Muscle cells have a whole set of tubes and vesicles that are filled with calcium. This this voltage change causes those calcium vesicles to release their calcium, and that causes muscle contraction. And we'll get into that later. But this is how we have the end game of this nervous system transmission. Just a little point here that makes it even that much more complicated and interesting. We can have what we think of in the biz as an excitatory signal, in that you are probably opening a ligand-gated sodium channel. But you also find in neurons, particularly in the brain, that you can have a ligand-gated chloride channel. And this, a ligand-gated chloride channel, now is going to act as an inhibitory signal. And as we learn more about the way the brain works, a couple of things come about. First is that there's a sort of threshold event that happens when you have particularly in the brain, when you have two neurons touch or synapse, you got the axon and then the downstream postsynaptic neuron. You have to have a couple of hits in order to have the next neuron actually fire, something called summation. So a single ion-gated or ligand-gated ion channel opening, a single axon, may not actually fire that downstream neuron. You may have to have a couple of them, because every neuron is going to have hundreds of axons touching it. I mean, it gets to be really complicated really fast. And so you have some kind of nervous system signal that is hitting this axonal gap, releasing neurotransmitters. That downstream cell has got the ligand-gated ion channels. Some of them are sodium channels, which are going to be excitatory. Some of them could be chloride channels, which is going to be inhibitory. And it's just sort of a game of what system kind of gets there first. You can begin putting more and more chloride in to make that neuron so it actually has a slightly more negative charge and it won't fire. You can put sodium in and pow, start the membrane depolarization that way. So it's not just an on-off. There's actually a lot of sort of in-between going on in the brain right now. And that's kind of a cool thing. Now, once the neurotransmitter dumps into the synapse, we have established the next movement, the next depolarization event. Let's just kind of think of it from that direction to keep it simple. And we now have to think about how do we clear the synapse? Because with membrane depolarization, we've got the system running down the neuron, and we hit and we dump out this neurotransmitter. That's going to cause this portion of the cell to start firing. If we maintain the neurotransmitter in the synapse, we can continue to keep that neuron firing. So that's not a very good control system. We have to have a way of clearing the synapse. And it turns out we've got a couple of different mechanisms for clearing the synaptic cleft of neurotransmitters. A classic one has to do actually with a lot of the way muscles are signaled to begin contracting. And the neurotransmitter you may have heard of called acetylcholine. So we've heard of choline before. It's one of the decorating molecules on phospholipids, and then it's got a 2-carbon acetate group on it. So acetylcholine is a good neurotransmitter. In the case of acetylcholine, the way the synaptic cleft is cleared is that as soon as the acetylcholine is released, there's another system being released as well, and that is a modifying enzyme that's known as, or it's called, acetylcholine esterase. Acetylcholine esterase is going to esterify acetylcholine and make it so it can't bind its receptor anymore. So it's effectively removing its ability to act as a ligand, and so you have this flush, and then immediately you have the esterase modifying it, so you just get a simple pulse. Just as a little interesting side note, there is a very potent inhibitor of acetylcholine that we know, and that is the alkaloid nicotine. Nicotine from the tobacco plant, actually probably a couple of other plants produce it. Nicotine is a brilliant inhibitor of acetylcholinesterase. I'm pretty sure it's a competitive inhibitor, but I don't know exactly. Regardless, nicotine at low dose is maintaining acetylcholine a little bit longer in the synaptic cleft. At high dose, it actually is keeping the acetylcholine in the synaptic cleft for a very long time, and at high dose, it's actually a really potent poison. At low dose, it just actually is sort of firing neurons a little bit longer, and you have all kinds of odd, subtle effects that are the result of nicotine ingestion. So if you're doing any kind of vaping, that's what you're doing, is in fact giving yourself an acetylcholinesterase inhibitor that is allowing the neurotransmitter to hang in the synaptic cleft a little bit longer. So that is one way to clear the neurotransmitters, by dumping the neurotransmitter out and then immediately dumping out a modifying enzyme. And so you're not really clearing it per se, but you're making that stuff out there inactive. Another way of doing this system is that you dump the neurotransmitter out, and then pretty much immediately afterwards, you start up a system that is able to actually use active transport and pull the neurotransmitter back in. So we have another way, and that is a reuptake system. So the neurotransmitter is dumped out, and then it's pulled back in. It's just a different way of doing it. Again, we've got some interesting pharmacological tools here. There's a neurotransmitter known as serotonin. And there are a class of drugs that are called selective serotonin reuptake inhibitors. And you may have heard of these as they're known often by their initials. These are known as a class of drugs that are called SSRIs, selective serotonin reuptake inhibitor. And what this is doing is inhibiting the reuptake of serotonin, keeping it in the synaptic cleft a little longer.
Why do we want to do this?
It serves as an antidepressant. This is what Prozac is, and Paxil, and pretty much most antidepressants that you may have ever heard of are generally SSRIs. There are some that are monoamine oxidase inhibitors, MAOIs. They are playing a sort of a nicotine-like role, and they actually are a little bit more aggressive. SSRIs are fairly gentle antidepressants. What they're doing is they're maintaining serotonin in the synaptic cleft a little longer. And not all neurons are using serotonin as their neurotransmitter, but those that do are part of the brain that actually begin to, they're often known as euphorics. They start to make you feel happy. Another example from this area here is, and it's a little bit like a different direction, instead of inhibiting the uptake, there are certain pharmacological agents that actually dump more serotonin in the synaptic cleft. Perhaps you have heard of lysergic acid, LSD. LSD actually acts to dump more serotonin into the synaptic cleft. So it's kind of doing the same thing as an SSRI, but just from a different direction. And LSD can generate all kinds of interesting changes in neurochemistry, and it also acts as a fairly good antidepressant. So, it actually has other issues in terms of hallucinations, which you wouldn't necessarily want an antidepressant to do. But after the hallucinations, you do have a significant sense of euphoria. Maybe in part because, wow, I'm no longer hallucinating. On the other hand, it also actually works that way. So there is a lot of monkeying around that you can do with your neurotransmitters and neurochemistry that adjusts the way the brain and the nervous system works. And so, this is an area of very rich study in terms of neurochemistry and the way the brain actually works. And, it's a pretty cool area of study. And then there's a lot of things going on here. So, actually, there's a really good fluorescent micrograph I want to show you. And how do cavities form? That's what we're going to talk about. But
So what's the advantage of keeping the, if they just stay there in the synaptic cleft, why do we want them to just sit there?
They are not going to hang there forever, but you are increasing the hang time in a sense. And it has some interesting effects. It does affect the way the neurons begin to fire. And so, the action is not real simple, and it's also not totally predictable, nor is it actually all that well understood. Part of this is developing drugs, and once you begin to say, okay, this is an enzyme we need to inhibit, like the reuptake system, or this is some sort of mechanism we need to inhibit, they begin targeting drugs that do that and then seeing what the effect is. Now, that's a good question. The answer is we don't have complete understanding of how increasing serotonin in the synapse is working, but it's causing certain parts of the brain to fire longer and harder, and that begins to change things up. There was actually a study now, it's been kind of an initiative of probably 5-6 years ago. We talk about the genome and the proteome, the whole thing of omics, of large-scale study. And there is an initiative, something that is called the connectome, which is to look to see how neurons are connected to each other. And it turns out that while there's a fair amount of diversity among our brains, there is very specific kinds of connection sort of classes that you can begin to make, and you can begin to map out these connections in the human brain. And you can begin to see that there are some individuals that have slightly different connections, and that ends up actually resulting in various pathologies. And so the connectome is a really cool thing to begin looking at. It's a very complicated story, and as we'll see in just a moment, I wish that, well, let's see, just to demonstrate it, and then we'll go backwards. This is a fluorescent micrograph in which we've got a neuron that is stained green, and there's a red stain that is antibodies that are binding to a protein in, I think, in neurotransmitter vesicles, or at the synaptic cleft, regardless. And check this out. Wait for it. This is a single neuron, and so you're looking at the dendritic side of it, and look at how many neurons are synapsing on it. Each one of these little red dots represents a synaptic connection. Each one is feeding excitatory and inhibitory signals into that neuron. And in response to all the stuff going on, that neuron is going to decide to fire or not fire. And this is one neuron. And each neuron has got hundreds of synaptic connections. So there's a lot of talking going on here. This is sort of a cartoon diagram of what we're seeing here, in that we've got the dendrite and the presynaptic nerve terminals of axons. All these different axons are hidden as one dendrite. And every single one of these has the ability to dump neurotransmitters depending on how it's fired. And so it is a massively complex web going on in the brain. Brain neurochemistry and brain interaction is really the next frontier in terms of understanding the aspects of physiology, but somewhere in here lies consciousness. This is how we're able to actually think about things and recognize our own self. So one of the things I think is very neat about this world we're living in and the science that we're studying is that we know pretty well the base cellular actions. But then you begin to expand it, and you end up with this next higher or multiple higher order levels of synergistic effects happening. I mean, it's a little bit like knowing that a computer works because of ones and zeros and off-on systems going into your, the central processing unit. All right, I know that, and yet my iPad still blows me away by the fact that, I got a computer sitting here. It is, at its base, very, very simple. At its base, this is ligand-gated ion channels. Then you begin to elaborate it, and all of a sudden, while you know the exact mechanism at the very base level, how this whole thing is working is a really complicated and unknown story. But I digress. So that's what I want to kind of finish with. I think and this is the last image in the book, just how complex this system can be. But let's go through a couple of other slides here. Hold on. Just a review here, because our next topic is metabolism, and depending on the time, I might back and fill and start it on Friday, or I might not. We'll see how we go. All right, so this is our axonal diagram, and what we were looking at was the cell body and all of these different axon terminals from a bunch of different neurons hitting that guy. This is all this information coming in, and then the information is essentially being sorted out by inhibitory signals and excitatory signals, until you finally go, okay, we're going to fire, and off the races. And then these guys are carrying out the same kind of thing. So the giant squid axon is the place where it's the organism that has been used for studying, and we have this kind of information, the action potential. It's actually being, it's done by using very small electrodes, and as we mentioned, whatever. Oh, well, this is the experiment. What you can do is, we know it's sodium because if you put the axon into a solution, so you've actually pulled it out of the animal and put it into a solution with different amounts of sodium based on the concentration of what's in the fluid that is, natively surrounding this axon. By reducing the sodium concentration, you actually can reduce that voltage spike, which is the pretty good evidence that says, yep, it's sodium going in. Another thing that was done is that rubidium will act like sodium in the ligand-gated ion, or the voltage-gated ion channel, and by taking the axon and placing it into a rubidium solution, you see that rubidium is going into the cell when the membrane depolarization event happens. And so this is the way neuroscientists were able to demonstrate that it is sodium rushing in that actually causes the membrane depolarization. So another thing is interesting to think about is that this voltage-gated sodium channel, it has this kind of plunger guy. And this is what's dropping open and allowing the channel to open up. There is a fly mutation known as shibiri. I'm pretty sure it's shibiri. It stands for, or it means shivering in Japanese, I think. And so these are flies that they quiver. And it turns out that they've got a mutation in their voltage-gated sodium channel. And as people began to manipulate the protein by doing recombinant DNA experiments, one of the things that was observed is that the full mutation is missing this portion of the protein, the little ball. What you can do is actually change the length of the chain, if you will. By shortening it, you cause the whole system to snap open faster. And so we could potentially manipulate sodium voltage-gated ion channels in people, changing the length of the chain and cause them to have a faster response time. So you might be able to have somebody that actually is able to work at a different clock rate. If you know this from computers, you can change the clock rate on your system and actually have processing events occur faster. No one's done that so far, but that is one more thing in my super soldier. All right, let's see. Yeah, we looked at this. Fine. All right, so this is now an electron micrograph of a series of axons that are synapsing with a cell. So this is the postsynaptic cell in the postsynaptic membrane, and this is the individual nerve synapses and the presynaptic cells. And so this here is the synapse. Not a whole lot of gap, but it's there. And these are all those neurotransmitter vesicles, all the synaptic vesicles, just waiting to flush in and bind to the receptors right here. So this is how the system is working. And a little higher mag there. Very cool. Yeah. So as we get into, or as the the voltage membrane depolarizations running through, we end up actually having in some of the neurons a calcium channel that opens up, and that causes the release of the synaptic vessel from its tether and allows it to now fuse the cell membrane, dumps out the neurotransmitters, and they can bind, and you're off to the races again. Let's see. Oh, so this is one of the ligand-gated ion channels that's using acetylcholine, and acetylcholine, when it binds, causes this thing to open up, and sodium rushes through. So just some examples of ion channels, and no need to memorize these, but it's just a range of things. We've got potassium leak channels, voltage-gated sodium channels, which we talked a lot about, voltage-gated potassium channels, which we talked about, voltage-gated calcium channels. These are at the end of the nerve terminal, and so this is what's actually triggering the release of those synaptic vesicles. The acetylcholine receptor at neuromuscular junctions. This is an excitatory constricting signal. Glutamate receptors. These are another excitatory signal. They're actually used mostly in the brain. The GABA receptor. This is an inhibitory system. GABA often will interact with so you have a GABA-gated chloride channel. And a lot of neurons have this as well, and this is a chloride going in, acting as an inhibitory signal. So rather than causing the voltage to go up, it actually further suppresses the voltage. Another inhibitory signal, mechanically activated cation channel in the auditory hairs. So that's how you can detect sound vibrations. All right, and here we are. Beep. And we finish. Oh, all kinds of weird-ass experiments looking at a blue light-induced neuron depolarization event. And I'm not sure what that mouse is doing, but... And what's this? Nothing. Good. In the slideshow. And... what time is it? 9:45. We really will stop, but I'm just going to do this myself to say our next topic. Hello, don't do that. Open up. Is how cells obtain energy from food. And we'll pick up on that on Friday. It's actually a really cool topic of how the food you're eating is being turned into energy. And I'll see some of you tomorrow. I'll see the rest of you Monday. Or no, hell, whenever I see you. I don't know. It'll be a while. Some of you I saw yesterday. I should stop recording. Hopefully we get some good comments.